Hello everyone, this is the first of a series of videos dedicated to mass spectrometry. In these videos we will be discussing the scientific basis of mass spectrometry and some of its strengths and limitations. We will also learn how to interpret mass spectra of simple molecules. And when I say simple, I don't mean they are easy to interpret. What I mean is that they are small, discrete molecules, as opposed to, for example, mass spectrometry of macromolecules, such as DNA or proteins, which you will study in future years. We will also be looking specifically at the combination of mass spectrometry and gas chromatography, which is called GCMS. And we will discuss the use of GCMS as a very powerful separation, identification, and quantification techniques. The topic is divided in several videos to make it easier to follow. So what is mass spectrometry? Well, a mass spectrometer measures the molecular weight of a particular sample with great accuracy. Even for very large samples like biomolecules, molecular weights can be measured to within an accuracy of 0.01%. So it's an extremely accurate technique. And for small organic molecules, the molecular mass that you can get from a mass spectrum, from using a mass spectrometer, can be measured so accurately that a mass spectrum, a high-resolution mass spectrum, can be sufficient to confirm the particular molecular formula of a compound. And for unknown substances, beside the molecular mass and the molecular forma, you can obtain structural information from the inspection of the mass spectrum. All mass spectrometers have these three distinct parts, which are the ionization source, the analyzer, and the detector. A very brief history of mass spectrometry, in case you're interested. It was first developed in the early 1900s, and these two gentlemen, J.J. Thomson and Francis W. Aston, both Nobel Prize winners, constructed together the first mass spectrometer. Those first designs were later improved by other scientists. For example, in 1918, Dempster developed a particular way of deflecting the ions, a magnetic deflection instrument, which is still used in its basis today. And he developed the first electron ionization source. And these developments introduce a huge improvement in the resolution compared with the initial spectrometer work that had been undertaken by Thomson. This is a schematic diagram of how a mass spectrometer works. Initially, the sample is introduced in the mass spectrometer, and perhaps it's in a gas form, and if not, it's vaporized. Once it's in vapor form, the atoms are ionized. And to make those neutral molecule into an ion, they are hit with high energy particles, which will knock an electron off those molecules and will make them from neutral into an ion. Besides knocking an electron from the molecule to make it into an ion, hitting it with high energy particles will make fragments of that molecule come off. So certain bonds will be broken. Typically the first to be broken are the weakest ones. So you will have a mixture in this ionization chamber of the original molecule in ionized form and fragments that have come out from the molecule, depending on the bonds that have been broken. All that cocktail of ions enters the spectrometer and it gets accelerated to provide all the ions with the same kinetic energy. Then it goes through this part, the deflection. Here, an electromagnet produces a magnetic field, and the different ions will be deflected because they get affected by the magnetic field, and they will be deflected with respect to their mass, or specifically their mass to charge ratio. So lighter ions will be deflected shorter distances. And by changing the magnetic field we are able to separate all those ions, so they leave the spectrometer one group at a time, depending on how much they have been deflected by the electromagnet. There is some sort of detection method, and finally, well, a chart recorder, or nowadays a computer. So as I mentioned in the previous slide, there is ionization process. In the first step, the atoms, the sample, your molecule, is ionized by knocking one or more electrons off the structure. And besides the molecule becoming an ion, there's also a breaking down of some of the molecules by through different bonds. All those ions are then accelerated, so they all have the same kinetic energy, and they go through a magnetic field, and which deflects them according to their mass, or specifically their mass to charge ratio. It's not only the mass, it's the charge of the ion. Typically, one electron will be knocked off, so the charge is 1, but in some cases you can see, for example, there are fragments corresponding to 2 electrons being removed. The lighter they are, the more they are deflected. So by changing that magnetic field, we are able to make the different ions leave the spectrometer one set at a time, depending on the mass or charge ratio. And finally, there is a detector in which we record the spectrum. All this process happens in high vacuum because the particles the ions are traveling so fast, and you don't want any particles of air or any substances in air to interfere with the path of those ions. So the whole system is kept under high vacuum. There are different types of mass spectrometers, and they get their name from the ionization method. So the method to make the molecule, the neutral molecule, into a charged ion. And it depends. For example, electron impact uses electrons, so the sample is bombarded with electrons. With fast atomic bombardment, FAB, the sample is bombarded with accelerated large atom ions. For example, sodium ions can be used. In the MALDI, matrix assisted laser desorption ionization, we use laser to produce the ions. Thermospray is the temperature that produces the ionization source. The different methods, the different ionization methods will be appropriate in different circumstances. Some of them, for example, electron impact, can fragment the molecule quite a lot and produce many fragments, which is okay for small molecules, but for example is not very helpful when you're studying large molecules, biomolecules such as protein or DNA, because you don't necessarily want information of very small fragments. What you want maybe is a larger fragment of the molecule, for example a small peptide fragments that will tell you information about the structure of those of that protein. So for that you prefer to use something like MALDI as a spectrometer to analyze biomolecules. So the different ionization sources have different applications depending on the size of the molecule you're studying and how delicate or how susceptible it is to breakage. And they also have different price points, and they are some of them can be extremely expensive because of this the ionization setup can be expensive to maintain. Why is mass spectrometry useful? Well, you can get quite a lot of information from a mass spectrum. First of all, you can know the exact mass of the compound, and if it's a high resolution mass spectrum, that will give you the elemental composition, so you can work out the molecular formula of the compound you're studying. Also, because you get fragmentation patterns, the molecule breaks down in the chamber and produces ions with fragments of the molecule. You can study that fragmentation pattern, and that will give you information about the structure of that molecule. And you can also look at isotope abundance, the presence and numbers of certain elements and their isotopes. And you can, for example, study isotopic enriched samples with, let's say, oxygen 16 or nitrogen 15. How do you interpret a mass spectrum? Well, you can use a database, and you will see, for example, when you look at GCMS, you can compare with databases, and it will give you a percentile match to your spectrum, or what that compound might be. And you can work out with exact mass the possible elemental composition. So once you know the exact mass, you can work out the molecular formula, and you can suggest how that how those atoms are put together in the formula. You can using also the fragmentation patterns to help you see how those atoms might be put together, or you can use a computer program, for example. What we are going to do in this module is interpret the spectrum. So a skilled scientist, a skilled chemist can interpret a mass spectrum and work out the structure of the compound by looking at the fragmentation patterns and what it says about the different combination of atoms that are present in that molecule. That will give you the structure.